But DNA is a synthetic substance which was first isolated by Frederick Mitchell 1869 from nucleus of pus cell. He called it nucleus. In eukaryotes, DNA occurs inside nucleus and two types of semi-autonomous cell organism, mitochondria plus chloroplast. DNA present inside nucleus is called cell nuclear DNA. The one present inside semi-autonomous cell organism are originally DNA. Nuclear DNA is rotated by histone protein to form chromatin fiber. Chromosome are condensed chromatin fiber. Original DNA is without histone protein. It is termed as neck DNA. Neck DNA also occurs in prokaryote. The DNA is without association with histone in prokaryote. DNA containing the organelle after nucleus is also absent. Nuclear DNA is embedded in the cytoplasm. It is often called chromosomal DNA. This DNA may be folded with the help of RNA and polymerase. Many prokaryotes also possess extranuclear or extrachromosomal smaller circular DNA segments called plasmids. Derivation of DNA structure. Chemical component of DNA has been discovered prior to 1952. The two lines of investigation help in derivation of DNA structure: Chargaff's rules and X-ray crystallography. Chargaff's rule. Through his work on estimation of various chemical components of DNA, E.F. Chargaff 1950 put provides certain generalizations. They are called Chargaff's rule. Purine pyrimidine occur in equal amounts. Purine found in DNA at adenine, guanine, pyrimidine of the DNA at thymine and cytosine. A T plus G equal to T plus C. Sugar, deoxyribose, and phosphate residues occur in equal number. Purine adenine is equivalent to pyrimidine and thymine. Purine guanine is equivalent to pyrimidine. Cytosine. Basic A plus G by C plus G is specific for a species. It does not change with age or tissue. The ratio can be used to identify species. Base ratio is the ratio between the sum total of adenine per thymine bases and the total of cytosine polygonals in the DNA sample. Extra crystal acetate duplication to the DNA by W3 as Bury 1940 indicated DNA is a polynucleotide where successive nucleotides occur at interval of 3.4 angstrom. Franklin 1952 discovered DNA to be Wilkins and Franklin 1952 obtained very fine X-ray diffraction pictures of DNA which were immediately made available to Watson and Crick. Passing together all the previous information, Watson and Crick 1952 came to the conclusion that DNA was made up of two anti-parallel helical chains which are held together by the hydrogen bonds created between their nitrogen vessels. Along with Wilkins, Watson and Crick were awarded Nobel Prize in 1966 of DNA. DNA is long double chain or duplex molecules form of millions of deoxyribonucleotides. Length of DNA is characteristic organism and is chromosome. Bacteria possess 1 by 4 has 5386 nucleotides. Bacteria for lambda possess DNA having 48502 base pair. Acetyl choline has a DNA consists 4.6 into 2.6 base pair. As single genome consists 3 of 23 chromosomes possess 3.3 into 2.9 base pair. In case of pneumonia, single stranded DNA molecules occur in some viruses. Qualiphage 5 into 1 by 74. DNA double helix at diameter 20 angstrom. The duplex is called platonymically is a right-handed manner just as a rope is twisted to form a spiral. Their coiling produces alternate major length 22 angstrom and minor length 22 angstrom groups. Each turn of a spiral has a distance of 34 angstrom. This length contains 10 deoxyribonucleotides in each sense for the average distance of the adjacent deoxynucleotide 3.4 angstrom. Four types of deoxynucleotide occur in DNA: DMP, deoxyadenosine monophosphate, DGMP, deoxyguanosine monophosphate, DCMP, deoxycytidine monophosphate, DTMP, deoxythymidine monophosphate. Each deoxynucleotide is further formed of three components: deoxyribose sugar, phosphoric acid, and nitrogenous base. Nitrogen bases differ in four types of deoxynucleotide. Two of them are 9-member double rings purines, adenine and guanine. The remaining two are 6-member single rings pyrimidine, cytosine, and thymine. Out of the three components of the deoxynucleotide, sugar, deoxyribose, and phosphoric acid form the backbone of DNA strand, while nitrogen base lies at right angle to it. The backbone is formed of alternate phosphate sugar phosphate residue. Nitrogen will project at the right angle to the backbone. The region of sugar residue. There are two strands of DNA. The nitrogen base of the two strands come to lie opposite each other and develop hydrogen bonds between the nitrogen base attached to OH of carbon 1 of deoxyribose sugar to glycosidic bond CNC by either side, and 1 in case of pyrimidine, cytosine, or thymine, or N9 in case of purine, adenine, or guanine region. Deoxyribose sugar is bonded to phosphorus to lead to 8 radical of is equal to OH of carbon atom. 5 phosphate radical is connected to residue sugar of next nucleotide at its carbon 3. Phosphodiester linkage to ester formation by same phosphate radical is created between sugar residue adjacent nucleotide in one chain of the DNA duplex. The last deoxyribose of one end has OH its carbon 5 free, while in the other chain the deoxyribose of the end has OH of its carbon 3 free. The condition of the other end of the duplex is reverse. The direction nucleotide chain is 5 to the 3 in the other. The need of the one lying timing of the other and cytosine one opposite guanine of the other. The opposite and complementary nitrogen will held together by hydrogen one two between A and plus 1 3 6 4 and 3 between C and G at position 1 1 2 6 6 2. Two types of forces stabilize duplex and hold the two DNA chains together. Hydrogen bonds between complementary nitrogen bases of the two chain hydrophobic interaction between nitrogen bases. The latter are also kept stacked inside the helix, while the polar groups are kept on the outside in contact with water. Base pair. It is the pairing formed with DNA in the double helix between pyrimidine of one strand and pyrimidine of the second strand. Base pairing is specific in with adenine lying opposite thymine and cytosine occurring opposite guanine. A proper base pairing is required for two purposes. The two DNA strands can remain exactly parallel only when the space between them remains uniform. Presence of two parents as base pair will make the DNA double helix wide due to bulging out, while two pyrimidines as base pair will make the duplex narrower. The space between the two DNA strands of the duplex 20 extend. The one available for the nitrogen base as about 11 and study can accommodate neither two parents nor two pyrimides. The space is present for one pyrimide and one purine with a small area in between for forming hydrogen, but only adenine, thymine, and cytosine guanine base pair have proper spatial arrangement and configuration to develop hydrogen bond. Two between A and T and three between C and G anti-parallel strand. The two strands of the DNA parallel but oriented opposite direction. Each strand anti-parallel. The 5 days end of one strand lies opposite 3 days end of the other. In one strand, the nucleotide running 5 days, 3 days direct one, and the other strand nucleotide occur 3 days, 5 days direction. The arrangement is useful in complementary base pairing and precise application of DNA. Sense and anti-sense strand. Out of the two strands of DNA duplex, only one carries the genetic information. It has 3 days polarity and is called template or anti-sense strand. The other strand is mirror copy with the polarity 5 days. It is called coding or sense strand. The coding or sense strand is no less important. It covers the nitrogen basis about anti-sense strand and protects the same from cellular biomolecule. The code present in it is similar to that of the mRNA except for the presence of thymine instead of uracil. Complementary. It is the occurrence of different specific nitrogen bases. The two strands of the DNA, A opposite T and C opposite G, a similar complementary occurs between the antisense strand and mRNA over its opposite way of DNA, A T opposite T and C opposite G. A sequence of C A G G T A C G A of templates T cell with a complementary G T C A T G C on the sense strand of DNA and sequence of GUCCAUGCU on mRNA. Linear and circular DNA. DNA is the one in which ends are breathed. In circular DNA, the two ends are covalently liquid. Circular DNA occurs in prokaryotes and semi-autonomous organelles, mitochondria and plants. DNA occurs in eukaryotic nucleus, some prokaryotes and occasionally cellular. Eukaryotic nuclear DNA is associated with histone. Prokaryotic DNA and organelle DNA are necked on without association with denaturation and denaturation. Exposed to high temperature acid occur, the two strands of DNA separate from each other. The denatural anomaly. It is caused by disruption of hydrogen bond between the nitrogen side of the two strands. DNA areas rich in AT generated more easily as compared to areas rich in CG. The former are called low melting area. It is because of the two hydrogen bonds in their base pair, while the high melting area of CG possesses 3 hydrogen bonds in the base pair. Denaturation carried out during in vitro synthesis of DNA. The latter are performed for the first time by Korenberg in 1950. The two strands of DNA separated by denaturation have the tendency to reassociate reassociation of separated or denatured or DNA strand is called denatured hereditary input. The antecedent strand of DNA carries hereditary information in the form of acetone and diacodone. Stone is that segment of genome which is expressed in the trace of the polynucleotide polypeptide. It is equivalent to a gene or hereditary unit. Stone contains a large number of nucleotides are in linear order. The three adjacent bases of nucleotide to determine incorporation of one amino acid in the polypeptide is called codon. The number of codon present in the system is equal to the number of amino acids present in the polypeptide formed by the latter. In each system often possess non-coding areas called introns. The non-coding area then called exons. System containing introns or non-coding areas are called split genes. As change in the sequence of nitrogen changes the codon of a system, this produces modified polypeptide RNA. Smallest segment of the genome which can undergo change is called mutation. Repetitive DNA, it is that part of the genome which contains the same sequence of nitrogen which is repeated more than once in the genome. The area with a long sequence of repetitive DNA is called satellite DNA because it's repeated out during density ultracentrifugation. It occurs especially near ends of chromosomes, telomeres, centromeres, pericentric, and other heterochromatic areas. Satellite DNA is of further two types: microsatellite with 1 to 6 base pair repeat units, and minisatellite with 11 to 60 base pair repeat units. Minisatellite DNA is hyper variable and specific for each individual. It is used for DNA fingerprinting discovered by Jeffrey et al. 1985. When Romy DNA, it is a basic sequence of DNA which reads the same order read from right to left or left to right. Polyneurobic DNA has a similar sequence in both the and different types of things recognized by the restriction interlinked DNA polymerase. The dictionary more than one form of the structure equal polymerase. DNA polymerase is the occurrence of different forms of repetitive DNA microsatellite, a single nucleotide and the scrub pigment length. A change in single nucleotide is found in human genome every about 1330 bases. Single nucleotide polymerase is responsible for the most of the genetic variation in human beings. Their number is believed to be more than 10 million already. More than 1 million have been discovered. Change in pattern of fragments produced by cleavage with the restriction enzyme is called restriction of fragment length polymer. Most of these sequences lie in between the genes and have no effect on phenotype. Both SNP and RFLP are used in genetic mapping. Salient features of DNA. DNA consists of two polynucleotide chains. They have two polynucleotide chains that run in antipolar fashion, 5 polarity in one and 3 polarity in the other. The double chain of DNA is helically twisted in right-handed fashion. Each turn of double helix covers a distance 3.4 nanometer. It is a roughly 10 base pair. The distance between the two adjacent base pair is therefore 0.34 nanometer, 1 nanometer, 10 to the 9 meter, or 1 millionth of a meter. The backbone of each polynucleotide chain is formed of repeated sugar phosphate residue. The DNA lies at the right angle one of the two chains so that their free ends come to lie opposite each other. The lineage of one chain lies opposite thymine of the other, while cytosine lie opposite guanine. A purine age lies opposite to a pyrimidine. Purine in a large size, while pyrimidine is small size. Their pairing keeps the width of the DNA double helix same. Hydrogen bases opposite chains are held together by the hydrogen bonds. There are two hydrogen bonds A and T. The three hydrogen bonds overlap between cytosine and guanine. The plane of the one base pair checks over the other along with hydrogen bonding staking provides stability to the helical structure. Hereditary information, all the hereditary information contained in DNA, it occurs in the form of systems or structure gene regulator or pressure gene is encoded in a regiment or a sequence of nitrogen bases. There is a reshuffling of DNA molecules during gamete formation and their fusion crossing occurs at the time of meiosis. Both the phenomena produce variation being about individually adaptive mutation. They are sudden inheritable variations which appear due to changes in the genetic material of DNA. Mutation or mother of all variation or a catalytic function. DNA is able to perform its carbon copy. The phenomenon is called application occur at the time of cell division. Heterocatalytic function, the DNA function assembly photosynthesis of the urine different types of RNAs. This phenomenon is called transcription control and metabolism. DNA controls cellular metabolism through transcription of selected RNA and subsequent synthesis of specific enzyme with the help of RNAs. Growth and differential growth, cell grow, divide, and differentiate from tissues and growth due to differential function of specific genes or system. Gene therapy, there is possibility of correcting if the specific heredity is ordered by changing the genes or their expression, then each individual happens as many subtle sequences. Studying these sequences DNA fingerprinting is used for the resolving parenthood disputes and the identification of individuals. The different development studying individuals are controlled by internal clock operating in DNA antisense therapy. It is the technique of silencing specific genes of individual pathogen with the help of RNA complementary to the nucleotide sequence. Central dogma. Central dogma basic formulation of molecular biology state that there is no way or unique direction of flow of information from master copy of DNA to working RNA and from working copy of RNA to building plain polymerase. Central dogma molecule was proposed as a Crick and it appeared as DNA and returned from DNA. The central dogma also written as follows: DNA replication, DNA transcription, mRNA translation polypeptide. In this dogma of molecular biology, genetically destroyed the four-letter language of DNA. The same is transferred from during transcription four-letter language of messenger RNA, but with two difference. Complementation in code level replacement of letter T with U. Commoner 93 has suggested circular flow of information wherein polypeptide formed during translation fused RNA, which in turn control information. RNA, DNA, RNA, polypeptide. In 1970, H. and D. Baltimore discovered retrovirus which contained RNA genetic material. The virus produced and reverse transcribed. The enzyme performed the central dogma reverse by translating the DNA over template of genetic RNA. The new central DNA then functions as the master copy, producing all RNAs through transcription and RNA control translation to synthesize polypeptide. RNA reverse transcription, DNA transcription, mRNA translation polypeptide process of information DNA from RNA is called reverse transcription or termination. S virus, HIV, human immunoretrovirus were reverse accomplished. The enzyme reverse now commonly used for synthesizing process the gene from mRNA in RNA, TMB, poliomyelitis virus, genetic RNA produces new genetic RNA, still copy DNA.